Welcome dear students. In this class we are going to learn about protein glycosylation. This is the continuation of my protein shortening lecture. In my previous first protein shortening lecture, protein shortening basic concept, I have discussed about how proteins enter inside the endoplasmic reticulum. Now in this class I will talk about how proteins are modified, specifically protein glycosylations occur. So in this class I will talk about three things. One is what is protein glycosylations, how this protein glycosylation occurred inside the endoplasmic reticulum, and what is the significance of protein glycosylation in protein shortening. So today's topic is protein glycosylations. Protein glycosylation simply means addition of sugar molecules. Addition of sugar molecules. Addition of sugar molecules to the protein. Okay. Now this protein glycosylation could be of two types. One is called O-linked, another is called N-linked. So this O-linked and N-linked protein glycosylation depends on the amino acid which are attached with these sugar molecules. In case of O-linked, the hydroxyl group, this is why it is called O-linked, because hydroxyl have oxygen, so this is O. So this hydroxyl group of serine or threonine amino acids are attached with sugar molecules. So a protein, let's assume this is a protein. So this is this protein. If these proteins have serine molecules in this position, here serine molecules or here threonine molecules or serine amino acids, if this protein have this serine, threonine or serine amino acids this side, then sugar molecule is added in this side, in this side, in this side. So when sugar molecules are added to the serine or threonine amino acids, then it is called O-linked oligosaccharide or protein glycosylations. And what is N-linked? In case of N-linked, there is a amino group present of asparagine amino acid. So you must remember asparagine. These amino acids have a amide nitrogen molecule. So for that reason it is called N-linked oligosaccharide addition or protein glycosylations. But here another information I want to share with you, that in case of O-linked, only serine and threonine is enough for sugar molecule addition. But in case of N-linked, not only asparagine but a tripeptide sequence is very important. So asparagine is present, then a X, that means any amino acid can be present, and then serine or threonine must be present. So in this protein where there is this tripeptide sequence are present, exactly this tripeptide, asparagine, any amino acid, serine or threonine, then this asparagine will be added with these sugar molecules. So there are two types of protein glycosylation can occur. So this is protein glycosylations. Now how this protein glycosylation occur inside the endoplasmic reticulum? So let's assume this is the endoplasmic reticulum plasma membrane. This is the cytosol. This is the cytosol. This is the ER membrane, and this is the ER lumen. That means endoplasmic reticulum lumen. So in case of endoplasmic reticulum, there is a lipid molecule present inside the plasma membrane called dolicol phosphate. So this is called dolicol phosphate. Dolicol phosphate. Now this dolicol phosphate is attached with different sugar molecules. You must remember the sequence. At first, this dolicol phosphate is attached with 2 N-acetyl glucosamine, then mannose residues, 9 mannose. So there are 2 N-acetyl glucosamine are present, then 9 mannose sugars will be present. 1 2 3 4 5 6 7 8, let's assume this is 9. So at first there is a O-linked and there is a N-linked. This O-linked oligosaccharide or protein glycosylation is very simple. Here 1 or 4 sugar molecules attached with a linear conformation. So O-linked oligosaccharide additions or glycosylations is very simple. But in case of N-linked, oligosaccharides add. So this is example of N-linked oligosaccharide. Okay. Now at first the proteins are not modified. At first the proteins are present inside the endoplasmic reticulum. This is the protein. And all the sugar molecules are actually attached with a lipid molecule called dolichol phosphate. There are 2 N-acetyl glucosamine. There are 9 mannose. This is the sequence. In case of plants, fungi, single cell eukaryotes or multicellular eukaryotes, this sequence is actually conserved. Now here 2 N-acetyl glucosamine, 9 mannose, and then 3 glucose molecules are attached. Let's assume these are the glucose molecules. This is green, 3 glucose. So these are the glucose molecule. So 3 glucose. So this is the sequence of the complete oligosaccharide that actually attached with the protein. Now there is an enzyme called oligosaccharide transferase. That enzyme cut here, and all the oligosaccharides are actually added to this protein. Okay, so let's assume this is the addition of this protein molecules or sugar molecules. So this is how the protein glycosylation occurs with the help of dolichol phosphate. And for the CSIR-UGC NET preparations, you must remember which are the carrier of this molecule. Like N-acetyl glucosamine are carried out by UDP molecule, mannose are carried out by GDP molecule, and glucose are carried out by UDP molecules. These are group B questions. Now this is the complete sequence of these glycosylations, and then these all sugar molecules are attached with this protein where there is a asparagine. This is N-linked oligosaccharide I am talking about, because O-linked is very simple. Now what is the significance of these glycosylations? Yes, there is 2 functional aspects of glycosylations. One is called protein folding, and another is called stability of the protein. So there are 2 functions of this protein glycosylation is there. The first is protein folding. How these sugar molecules when attached in the protein helps in the protein folding? Yes, there is a quality control mechanism present inside the endoplasmic reticulum. You must remember, at first these sugar molecules are attached with the protein. Then there is a mechanism and there are some enzymes are present like glucosidase. That glucosidase enzyme cut these 3 glucose. Glucosidase enzyme cut these 3 glucose. Now there is no glucose present in this oligosaccharide. This oligosaccharide is present in the protein. Okay. Then another enzyme is present called mannosidase that also cut a mannose. I will talk you what happens, because now the number of mannose become 8. At first it was 9. Now 1 mannose is cut or removed. Then the mannose number becomes 8. When this oligosaccharide is 2 N-acetyl glucosamine and 8 mannose, that type of protein, because this oligosaccharide is attached with the protein I am showing here, but it is also present in this protein. A protein that have 2 N-acetyl glucosamine and 8 mannose that are actually allowed to come out from the endoplasmic reticulum to the Golgi body. You have to understand this. This is the license. So when a protein is properly folded, then this modification occur. 2 and 8, there is no 3 glucose and 1 mannose is cut out. Then this protein is allowed is allowed to come out from the endoplasmic reticulum to the cis-Golgi network. Then this protein can go through this process, secretory pathway. But if there is any problem in this protein folding, then the modification occurs in a different way. At first 3 glucose are cut out, then another glucose is actually added. At first 3 glucose are cut out, but if there is any problem in the protein folding, then another glucose will be added, and there is also 9 mannose are present. But in these situations, when there are 2 N-acetyl glucosamine, 9 mannose and 1 glucose, 2 molecules actually attach and actually scan the protein called calnexin and calreticulin. These proteins are present in the endoplasmic reticulum. This protein actually attach with these sugar molecules and actually help in the retention of the protein inside the endoplasmic reticulum. Okay, so you must remember that if there is any N-acetyl glucosamine, that is number 2, and mannose is 8, then this type of protein are actually properly folded. They can come out to the cis-Golgi network and go to this secretory pathway. But if there is any problem of this protein folding, then 1 glucose molecules are added. So there will be 1 glucose molecule. 1 glucose molecule will be added, and mannose will not be cut out, so there will be 9 mannose. Then calnexin and calreticulin will scan the total protein, and they help in the retention of the protein inside the endoplasmic reticulum. They will not allow this protein to go to this cis-Golgi. Now, if they cannot perform folding, if folding not occur properly, then mannose trimming also occur. Mannose trimming, it is called trimming. Mannose trimming occur, and after that glucose molecules also removed and the mannose are trimmed, and it becomes only 5 to 6 mannose. So mannose numbers become 5 to 6, and in that situations another protein actually attach with this sugar molecule or protein called OS9, and this molecule helps in the degradation and actually transport out this protein outside the endoplasmic reticulum. So OS9 protein attach with this type of oligosaccharide and can sense that this protein is actually misfolded, not properly folded. Then it helps in the transport out of this endoplasmic reticulum protein outside the endoplasmic reticulum to the cytoplasm, because that protein go through proteasomal degradations. Okay, so this is the protein folding and stability. It has been found that those proteins are not glycosylated, they are less stable than their counter partner, those are actually glycosylated. So you have learned here 2 important things: O-linked, N-linked, how these oligosaccharides are added, and what is the complete structure of this oligosaccharide. There will be 2, 9 and 3 glucose. If there is 2 N-acetyl glucosamine and 8 mannose, they are allowed to go outside this endoplasmic reticulum to cis-Golgi. If there is any problem in this folding, then calnexin and calreticulin actually attach with these sugar molecules, and then 1 glucose is added. If they cannot do the folding, then trimming occur, glucose are trimmed, mannose are trimmed, and this OS9 protein attached with these molecules and actually release this misfolded protein outside the endoplasmic reticulum. So by this way, by this protein glycosylations, misfolding or protein folding actually checked. This is quality control, and the protein is actually very stabilized in when they are glycosylated. So thank you for watching this video. If you like my video, subscribe this channel.